Right, so we are live and we will get started within next minute or so. All right, so everyone, pleasant good evening and welcome to this session on computer aided drug design. So, what is it which we are going to discuss today? How is it different from what we did few months back? I suppose almost 6 7 months back we did do a course on computer aided drug design. But how was that different from what we are going to do today? How specific we are going to be today about Pymol and Autodoc. So, what is basically Pymol? Just a brief about that, maybe a brief about Autodoc, how both of that things work. Most important thing, what is the application of computer aided drug designing or Pymol or Autodoc in our pharmaceutical industry? I hope all the BPharm students are already aware about the fact that our syllabus now officially has computer aided drug designing as a part of semester 8 syllabus. So, very obviously, there are a lot of things which are going to be new, which many of us are not aware about, which has a good scope in the industry. One by one we'll discuss about all those aspects. We'll have a lot of questions to be discussed which we can surely take up at the end of the session. Meanwhile, whenever between, the trainer will address that particular doubt as well. Today we have with us a renowned person who has been working on this stream of computer aided drug designing almost since last 5 to 6 years minimum, I would say. He has worked on different softwares like Schrodinger and all as well. He's a PhD candidate in medicinal chemistry, an alumnus of NIPER, and presently associated with one of the MNCs. So, welcome, sir. Welcome for this session on a Sunday evening where we are here to discuss about the advances in computer aided drug designing and, of course, what are the industrial applications. So, over to you, sir. We can get started. Your screen sharing has gone off.
Yeah, thanks, Ramesh. Just 1 second. Just let me know if things are working. Yeah, is my screen visible now?
Just a minute. Yeah, we are back in place.
Okay, great. Thanks, Ramesh. Thanks a lot, and welcome all of you. Welcome to this introductory session on computer-aided drug design. So, as I'm not sure how many of you over here are, you know, accustomed or have some idea about computer-aided drug design. So, those of you who have joined us live, could you just let me know how many of you have a basic idea of computer-aided drug design? Can you please let us know in the chat section? If
Also,
you
at
have
the
some.
same time, if you have heard something about CAD and it's only partial and you want more clarity, please address it in the chat box. So we'll be more than happy to address the doubts.
Exactly, and I mean, it will give me an idea of from where we can start off, and what all information would be valuable to you, because honestly, there is so much information that can be given on computer-aided drug design, and of course, you will learn it if you do end up joining the course as well. But just to okay, so you have it just okay, a very basic idea, I would say, as I can see from the chat section. All right, great. So let me just give you some idea of what we are going to cover over here. Okay, so we are basically going to talk about the scope of computer aided drug design, the utility, and whom is it for. Okay, like who exactly should think about having a career in computer aided drug design, and of course we'll be talking about what exactly it is. So just to highlight the importance, the 2021 breakthrough of the year, as you can see, let me just switch on the laser pointer. So as you can see, the 2021 breakthrough of the year, which is basically given by Science magazine. So Science magazine is one of the most prestigious and most popular science magazine. So it was given to AI powered predictions to show proteins. So basically, that the sequence of proteins. Okay, so basically each and every protein is made up of different amino acids, right? A sequence of amino acids. So to understand 3D structure of proteins, we basically need the crystallographic structure. Okay, what exactly, you might ask, is crystallographic structure? Well, crystallographic structure is basically, it's a technique. You might have heard about x-rays, right? So using x-rays, using x-ray crystallography, it's a technique, we find out the 3D structure of the protein. Okay, again, there might be a question in your mind that what exactly is the role, or why do we need the 3D structure? I will address that question very soon. Okay, so X-ray crystallography is used to predict the 3D structure of the protein, but this technique is very cumbersome and it is very, very expensive. Okay, because you have to crystallize the protein, and crystallizing such a huge molecule, protein generally are very huge in structure, right? They are made up of thousands or hundreds of amino acids. So crystallizing them is not very, very easy. So what has happened in this year 2021 was that AlphaFold, okay, which is basically you can say algorithm, artificial intelligence algorithm, which was developed by DeepMind, which is nothing but a offshoot of Google. Okay, so you can say it's somewhat a company of Google, which came up with this artificial intelligence tool, which can predict the structure of the protein, the 3D structure of the protein from its amino acid sequence. So that was the one of the biggest breakthroughs, and that has got a lot of people interested in this whole domain of using a computer, or using artificial intelligence, or using machine learning in the field of drug discovery. Okay, so this was, you can say, like a accelerator. Okay, even before this there was a lot of interest in this domain, but this just accelerated the whole phenomena. So there is a lot of interest in this, and there are a lot of, you can say, now positions in the industry as well as in the academia related to computer aided drug design. Okay, so let me just 1 second. Yeah, so let's talk about what exactly is computer aided drug design and what exactly is its scope. Okay, so if you talk about computer aided drug design, I'll come to the scope part later, since many of you are not familiar with computer aided drug design, what it does. So as the name suggests, computer aided drug design. So basically, when you are making a molecule, which will potentially act as a drug. So most of the, you can say, drugs that are there in the market, you know, of course they are made up of atoms, but there are different varieties of drugs. Okay, so for example, some of them are small molecules. Okay, small molecules means their molecular weight is less than 500. Okay, generally, I mean, it can be more than that also, but generally you can say small molecules are like, molecules with your molecular weight less than 500. So that is one class of small molecules. Okay, then there is something called as peptides. Okay, so peptides are nothing but small sequence of amino acids uh, that basically are bonded together through a peptide bond, right? So we have peptides, and then we have different kinds of other things like antibodies are there, and which are like macromolecules. Okay, so computer drug design is utilized in all of them. Okay, it can be used to design macromolecules, it can be used to design peptides, and it can also be used to design small molecules. But majority of the scope of computer aided drug design has been in the domain of what you call small molecules. Okay, now if you, as you can see on the screen over here, this is a typical pipeline for a drug to be entering into the market. Okay, into basically like to your homes or to your pharmacy, whatever it might be. So this is a typical process. So this stage till here is called preclinical. Okay, so till step 4, this is called preclinical stage, and after that there is the clinical stage. So the utility of CAD is in the preclinical stage. Okay, how it is utilized and what exactly it is, let us come to that. So basically, the first step when you talk about the preclinical stage, the first step is, let's say there's a disease. Okay, and what happens is, let's say that uh, that what the biologist would do. Okay, so a company would have biologists, or even in in universities you have biologists, right? So what their role is, they need to find that, okay, if Alzheimer's, okay, if a person is suffering from Alzheimer's, what all proteins are, let's say, elevated. Okay, what all proteins, so proteins play different functions in our body. So the role of this biologist would be to find out that, okay, if a person is suffering from cancer or some particular cancer, let's say pancreatic cancer, or let's say if a person is suffering from Alzheimer's, what are some common proteins that are found in elevated amounts, either elevated or depressed amounts. Okay, depending upon some proteins might be elevated, some proteins might be in reduced form also. So let's say the biologist will find, okay, there are certain types of proteins which are having elevated levels. Okay, some kinases. So kinase is a class of protein, and let's say this protein is some kinase, some particular kinase is found in elevated amount. So this then what will happen is the biologist will do knockout studies. Okay, what exactly are knockout studies is that there are some genetic manipulations that are done so that that protein can be totally knocked out. Okay, basically that protein will not be expressed in, let's say, some mouse model. Okay, and what they might find is that when you knock out this protein, okay, when you remove this protein, suddenly the you can say the disease either reduces or completely vanishes. So one can validate that, okay, this particular protein is essentially causing this disease. Okay, so what a biologist will do is first of all identify which protein is having elevated amounts, and then among those elevated proteins, when they do knockout studies, that is, they eliminate that protein completely. And if they find reduction in the disease, or if they find completely removal of the the symptoms of the disease, then one can say, okay, this is a validated target. Okay, so that's what it means by target identification. Target is nothing but the protein of interest. Okay, so and validation is when you do knockout studies, when you remove that protein and the disease reduces or completely vanishes, then you can say that particular protein has been validated. Okay, so it's called target of interest or protein of interest. So once the biologist has done that, then comes the chemistry part, the chemistry, or the you can say the computer aided drug design part. So now that you know that there's one particular protein that is causing this disease, now how do we end up designing molecules? What kind of molecules we can design? So that is a big question. Okay, that what do we do now, right? So there are two ways by which this hit generation step can be done. So there are large pharmaceutical companies and there are small pharmaceutical companies. Okay, so one way is that if we have the 3D structure of the protein. Okay, so why do we need 3D structure of the protein? Because when you develop molecules which will, let's say, inhibit the function of the protein. So when the biologist did the knockout studies, he or she identified that, okay, that if we remove this particular protein, then it can eliminate the disease or it can reduce the disease. So we need to find some molecules that will inhibit the function of that protein. Now, to find these molecules which will inhibit the function, we need to know the 3D structure of the protein. Because what happens is when you look at the 3D structure, I will show you if I have. Yeah. So let's say this is one of the 3D structures of this protein. So this over here is a pocket in the protein. And this pocket in this protein is actually responsible for, let's say, functioning of the protein. Okay, so this is, let's say, a protein which is found in elevated amounts. So what we need to do is we need to identify the 3D structure of the protein so that we can see some small pockets which might be functioning, which might be acting as like which will be the places from where the protein will function. Okay, so let's say there are certain sequence of amino acids in this pocket which are leading to the function of the protein. So if we make a small molecule, or if we design a small molecule which goes and fits inside this pocket, okay, if it goes and fits inside this pocket, we will come to know, okay, that this might be a particular molecule which might go and fit in the pocket and thereby inhibit the function of the protein. So that is why the 3D structure is very, very important, because without the 3D structure, you can say we are blind. Okay, we don't know what kind of molecule to make, because if we don't have the 3D structure, we don't know what are the pockets. Okay, what are the pockets in which we can, from which we can design the molecule. So depending upon the shape of the pocket and depending upon, let's say, there are other factors also. It's not just shape of the pocket. Like you might have heard about the lock and key mechanism or lock and key model. Okay, where the small molecule acts as a key and the protein acts as a lock. So the key fits inside the lock. So that is a very simplistic model, but there are other parameters also that you need to take into account when you're designing the molecule. For example, you need to take into account the polarization. Okay, like what kind of amino acids are there, right? For example, if arginine is there, glycine is there, valine is there. Okay, what all kinds of amino acids are there, and what is their polarity, whether they're negatively charged, whether they're positively charged. So you need to make molecules in such a way that the positively charged center of the protein should interact with the negatively charged center in the small molecule. Okay, so there are a lot of factors that need to be optimized for us to obtain a very good molecule. All right. Anyway, so like I said, so in the hit generation step, we need the 3D structure of the protein. And that is where this discovery was very, very important. Because even if the 3D structure of the protein is not available, okay, still we can have some kind of prediction. And this prediction is found to be very, very accurate, by the way. Okay, so we have a so we have the 3D structure of the protein. Even if the experimental structure is not available, we can make use of the artificial intelligence structure. Okay, anyway. So in this hit generation step is where the computer aided drug design starts coming into the picture. Okay, because now you have the idea of which pocket you need to inhibit, or which pocket the small molecule needs to fit. The pocket, by pocket I mean the pocket in the protein. Okay, a binding site in the protein. So we have a general idea of what is the structure of the binding site. So accordingly, now we need to make small molecules that will go and bind inside the binding site. So as a computer aided drug design expert, what your role would be is basically to you can say give the medicinal chemist, so medicinal chemist or organic chemist who will synthesize the molecule, give them an idea, okay, that what kind of structures might be useful in inhibiting the function of the protein, or what kind of molecules would be useful in basically going and binding to the protein. Okay, so you will basically run your studies by whatever tools you might have, and thereby give the medicinal chemist some idea, okay, as to what molecules he or she needs to start off or needs to synthesize. Okay, then based on your study, the medicinal chemist will start synthesizing the molecules. Okay, so once these molecules have been synthesized, they will give you some feedback, of course, because they will be given to the biologist who will do the inhibition studies, and they will tell you that whether the protein is being inhibited or not. Okay, or what kind of molecules are inhibiting the protein and what kind of molecules are not inhibiting the protein. So based on these experimental results, what you would do is, again, you will use your tools. So computer aided drug design experts have certain tools. So again, you will use these tools and you will come to a conclusion, okay, what kind of molecules will better inhibit the protein and what kind of molecules will not inhibit the protein. And then again, you will give suggestions to the medicinal chemist. So the medicinal chemist will again synthesize, and like that, from the hit generation step, you will go to lead generation. Okay, now you might be questioning, what is this hit generation and lead generation? So hit generation basically means that when there is a certain you can say cutoff that is set generally. Okay, whenever you are targeting a particular protein, there's a certain cutoff that is set. Okay, so when we talk about hit generation step, generally we say that, okay, 5 to 10 micromolar. Okay, if we get some activity between 5 to 10 micromolar, okay, then we can say we have reached the hit generation step. Okay, now what is this 5 to 10 micromolar? So you might have heard about molarity, the concept of molarity. So basically this 5 to 10 micromolar, what it means is that if we have 5 to 10 micromolar amount of the drug, okay, or whatever molecule that you have synthesized, if we have, let's say, about 5 to 10 micromolar, okay, or let's say, to be exact, we have 5 micromolar. Okay, we have a value of 5 micromolar. So when we say we have a value of 5 micromolar, that is called the IC50. Okay, inhibition concentration of 50%. So what that means is that if there's a protein, and if we are able to inhibit 50% of the function of the protein at 5 micromolar concentration of the drug, I'll again repeat, if we have a small molecule that we have synthesized, okay, that can inhibit the function of the protein up to 50%, okay, at 5 micromolar concentration, then that is basically called the IC50 of the drug. So the lower the IC50, that means the lesser the drug you require to inhibit 50% of the protein. Okay, so lower the IC50 value, or lower the, you can say lower the IC50 value, that means better is the drug. Okay, so in hit generation, basically you try to find somewhere between 5 to 10 micromolar. Okay, again, this is a rough idea I'm giving you. Okay, it could be higher also. So it depends on individual to individual what kind of concentration they might want for the drug. Okay, it could be 5 micromolar, it could be 10 micromolar, it could be 15 micromolar. Okay, but in general, very good drugs, okay, very good drugs will have inhibition in nanomolar. Okay, so micromolar is what? 10 to the power minus 6. Nanomolar is 10 to the power minus 9. So generally, many good potent drugs have values in nanomolar. So in hit generation, one more thing you need to understand is that you don't take into account much the toxicity of the drug or the metabolic stability, because see, this hit generation, whatever you are doing, no, this is in vitro. Okay, so there's a concept which is called in vitro, and there's a concept which is called in vivo. So in vitro is when you are doing some study on external cell line. Basically, you're not doing it inside a, you can say, either a human or an animal. Okay, you are not doing any of these studies inside a human or an animal. Then basically you call it in vitro. Okay, you are doing it on external cell lines. So this hit generation study generally is done on external cell lines. So of course, the toxicity of the drug, the metabolic stability is not that you can say important when we are going for the hit generation step. Okay, once we have found a hit, okay, once we have found a molecule that is acting that is inhibiting the protein's function at 5 to 10 micromolar, then we will go to the lead generation step. Again, here also your computer aided drug design expert is very, very important, because when you're going from hit generation to lead generation, that means you want a molecule which has potency in nanomolar. So from micromolar, now you're going to nanomolar, that is 10 to the power minus 9. So you need to improve the potency. So again, over here, the computer-aided drug design expert will go to his 3D structure of the protein and see that, okay, how this small molecule that was acting as a hit, now how is it basically going and binding inside the protein, how it is binding to the binding site of the protein, and what all modifications can we make in this hit molecule in order to improve the potency. So again, this is where you will come into the picture. Okay, that you will dock that molecule inside the protein, inside the binding site of the protein, see what orientation it is having, and then from there you will say, okay, let's make certain modifications which will improve the potency. Okay, now how do you improve the potency as a computer aided drug design expert? How will you improve the potency? So you have to basically locate certain interactions which basically improve the binding of the small molecule to the protein. Okay, I'll again repeat. What you want to do is you want to find small molecules which or you want to find certain sites in the binding site which will improve the interactions of the small molecule. Okay, so it could be different kinds of interactions like hydrogen bonding. I'm sure many of you might have heard of. Then there is something called as pi-pi stacking interactions. Basically, what happens if there is, let's say if there is tryptophan. Okay, tryptophan is there, or let's say phenylalanine is there. So phenylalanine, has a side chain in which there is a phenyl. Okay, and let's say in your molecule also there is a benzene kind of structure or a phenyl kind of structure. So when two phenyls are very close to each other, they show something which is called stacking interactions. So you might have seen like, if you take a pile of roti and you keep on top of each other, okay, or if you take a pile of pancakes, you stack them against each other, right? You stack them. So similarly, the phenyl rings will also stack against each other, and that is called the pi-pi stacking interaction. So basically, you need to identify such interactions which will improve the binding of this small molecule to the protein. Okay, so that is where your utility will come into the picture. And like I said, you will also have to factor in metabolic stability, or let's say solubility, because the drug needs to be soluble also. So there are a lot of parameters that you would need to optimize when it comes to this stage. Okay, another thing that you would not want to do is, for example, over here, as you can see, this is the binding site of the protein, right? This over here is the binding site. Now let's say this is the small molecule that you have designed, but this small molecule has some sort of toxicity, or let's say it has some sort of solubility problem. Okay, it has some sort of solubility problem. This drug is not soluble. So now you need to know about certain modifications which will improve the solubility. So as a computer aided drug design expert, your job is not only just to do some kind of studies, like you virtually do some studies and that's all. Okay, that is not your job. You also need to have a lot of knowledge of medicinal chemistry, that what modifications might lead to improvement in solubility. Okay, or what kind of modifications lead to improvement in metabolic stability. So it's not just that you are just doing some computer studies. Okay, you need to have a very good knowledge of medicinal chemistry also. Okay, and trust me, drug discovery in general, okay, I'm not singling out medicinal chemistry, but drug discovery, I can honestly tell you, in general, is also very, very interesting. Okay, it's very interesting how things work, and there are a lot of unknowns that people are still trying to optimize. So there's a lot of scope in each and every domain of drug discovery. All right, anyway, so like I was saying, so let's say I want to improve the solubility of this molecule. Now if I have done certain studies, okay, like one of the most basic studies in computer aided drug design is called molecular docking. Okay, it's called molecular docking. So let's say I've done docking of this molecule inside the binding site, and I know that this part of the molecule is very, very important. So if I want to improve the solubility, I would for sure know that I cannot make any modifications over here, because this part of this molecule is very, very important for showing the potency. Okay, so what I will do is that I will make some modifications on this phenyl ring, because as you can see over here, this phenyl ring is actually some in a region which is called solvent exposed. Okay, nothing complicated. It basically means that this is the binding site of the protein, and so basically protein is what it will be inside us. Okay, it will be inside the human body, so it will be, so what is a solvent in the human body? It's basically water, right? So it will be in the solvent exposed region. Okay, so this is where the water molecules might be present. Okay, so if you make any modifications over here, you can see that this is the binding site. So this site is very, very important. But if we make any modifications over here, that will not lead to any change in the potency of the molecule. So what we can do is, to improve the solubility, we can make some modifications on this phenyl ring on this side. Okay, so we will see that the potency is not dropping, but the solubility might be increasing. Okay, so this is where also a computer-aided drug design expert will come into the picture. So to improve the solubility, the metabolic stability, right? So majority of work that has been done by, or where the computer-aided drug design expert is required, is in the preclinical stage. Up to this stage, in clinical trials, there is not much that has been where a CAD person will be coming into the account. Okay, where a CAD expert will be utilized. Most of the CAD work is done till this stage, the preclinical stage. Okay, so this is where your expertise or your domain will be tested. It will be tested in the preclinical stage. Okay, is this clear to everyone? I hope if you have any doubts still, like in whatever I have questioned or I have basically demonstrated, do let me know, else we can move forward. Yeah. Okay.
After, sir, sorry, there was a question in the chat box which says, is PIC 50 or both are same? I guess there was some stage. Hit optimization is same as PIC 50 or something of that sort by
Is it
Abhimanyu.
same as, or both are same? Okay. No, PIC 50 is just it's just a value. So I think this question was by Abhimanyu. So Abhimanyu, PIC 50 is nothing but a value. Okay. And hit optimization is basically, yeah, I mean, to some extent, whatever you are asking, Abhimanyu, that makes sense. That in hit optimization, there are different goals. Okay. So PIC 50 is not, it's just one goal. Okay. So PIC 50 is nothing but, a different form of representing IC 50. Okay, so that is just one of the goals. So that depends on industry to industry what kind of goal they want to have. For example, Abhimanyu, just to give you an idea, you would see a lot of companies are working in the field of oncology or cancer. Okay, so the reason behind that is because in cancer or in oncology, in the field of oncology, you can have a very high tolerability to toxic drugs. Okay, what I mean by that is, see, all molecules will be toxic also in some sense. Okay, they will have some kind of toxicity associated with it. But in the case of cancer, or in the field of oncology, you can have more toxic molecules also. Okay, I mean, because cancer is so aggressive, so the regulatory bodies basically that regulate the toxicity of drugs, that how much toxicity is fine, they kind of like do not have a problem with the high toxicity of the drug also. Okay, so it might be curing, but again you would see a lot of side effects in cancer drugs. That's because their safety tolerance is very high. Okay, the regulatory bodies have kept a very high safety tolerance, and by high safety tolerance I mean which basically, even if they are toxic, but if they are not very toxic, they are still approved. Okay, but for certain other diseases, okay, like for example inflammation, okay, if you want to make anti-inflammatory drugs, their toxicity has to be very, very low. Okay, so it depends on what kind of disease you are trying to basically eliminate. Okay, so there also hit optimization varies. So for example, if you are a company which is working in the field of anti-inflammatory drugs, okay, then in hit optimization step, apart from this IC50 value, you also need to keep in account that the toxicity is also very less. Okay, because when you go from hit generation to lead generation stage, you improve the properties. Okay, so you cannot have a very toxic molecule also in the hit optimization step. Okay, so there are a lot of other factors. So IC50 is just one of the factors that is to be considered in the hit optimization step. I hope, Abhimanyu, I have made myself clear. And as I can see, there are not many questions as such. So I guess I would continue. Uh,
Sure.
okay. Yeah. Okay. So talking about the scope of computer-aided drug design. Okay. So I'll be talking about this its scope in industry and academia. And apart from that, there are two places where you can actually utilize CAD. Okay. One is the application part and one is the development part. So, like I said, if I go back to this, uh, so when you talk about the hit generation or the lead generation stage, okay. Now, in this, I said that you utilize different tools. Okay, so utilizing the tools, there are different tools. Okay, like I mentioned over here, molecular docking is one of the domain, molecular dynamics, QSAR, homology modeling. Okay, I'll not go into the into like what exactly they do, but there are different basically fields in the computer-aided drug design. Among these two are very, very popular. Okay, molecular dynamics and molecular docking, these are very, very popular in the field of pharmaceuticals. Okay, now if you talk about the application part, so application is, of course, as you can understand, somebody has made the tool. Okay, somebody has made this tool which you will be utilizing in the field of drug discovery or in other field. For example, in formulation also, now you will see computer aided drug design is being used. Okay, to basically, it's basically a part of material chemistry, or formulation, but still CAD techniques can also be used to develop, to basically understand what kind of formulation might work for a drug. Okay, so CAD is not just limited to hit generation and read optimization. Like I said, it can also be used in formulations, and other areas also, like for example in toxicity studies also. Okay, anyway, so that is all a part of application, that somebody has developed a software and you are just utilizing the software. And like I said, it is not just about utilizing the software. You need to have very good knowledge of medicinal chemistry or the drug discovery process in general. Okay, when you might initially enter this field, only a small idea of medicinal chemistry is good. But when you have some experience, or when you gain experience in this field, your knowledge should not be only limited to medicinal chemistry. You should also have knowledge of, let's say, formulations. So that will automatically, your knowledge will increase as you spend more time in this domain. Okay, so all that is a part of the application. Then there's another, actually more rewarding, I would say, field, which is development. Okay, application is also rewarding, no doubt about it, but as of now, if I talk about the current scenario, development is very, very rewarding. So in development, what you're doing is basically you're building the software from scratch. Okay, so basically you are developing this computer aided drug design tools. As an application scientist, not an application scientist, as a scientist, you are applying, either you are applying these tools of computer aided drug design, or you're developing them. And the development field, like I said, is has picked up a lot because of lot of collaborations between different software companies and different drug discovery companies. Okay, so Microsoft collaborating with big pharmaceutical companies, even Google is also collaborating with pharmaceutical companies. So as a developer, what you would be doing is, of course, you would have some knowledge of computer aided drug design, but at the same time you should also have knowledge of coding. Okay, so it could be either developing a software, or it could be in developing a artificial intelligence tool or a machine learning tool. Okay, so as a developer, you can also enter the field of computer aided drug design. So if coding is something that you find very, very you can say interesting, okay, or if you wanted to do coding, or if you had some interest in computer science, and at the same time you had some interest in the field of medicinal chemistry, then definitely you can look into the development side. Okay, but if you're not interested in coding, so honestly speaking, I am not very, very I was not very inclined towards coding. Okay, I like the field of drug discovery, and coding is something that I do as a hobby. Okay, so it's not something that I'm really passionate about. So I definitely prefer the application field. But if you really enjoy and are passionate about coding, and at the same time you like pharmaceutical or drug discovery as a process, then you can also look into the development field. Okay, now if I talk about industry or the academia, of course there are more opportunities. If you only talk about computer aided drug design, just this domain of computer aided drug design, then industry has a lot of scope. Okay, both in the development aspect and the application aspect. Okay, so there's a lot of scope in the industry. Academia also has scope, but comparatively the scope is less. You might see a lot of publications in the field of academia related to CAD. But if I talk about the jobs, okay, so in academia, like PhD, MSc can be done, okay, in the field of computer aided drug design. But the kind of jobs, faculty jobs that are there are comparatively less right now. Okay, the more there are more jobs in the industry. Academia is generally, you can consider, a little slow to adapt to new things. Okay, so academia will take time for, for understanding, or basically, utilizing the utility of CAD. But industry, there are a lot of opportunities right now. Okay, especially if I talk about right after, because of COVID, there has been a lot of interest that has been generated in the field of pharmaceutical or healthcare. And because of that, you would see that more opportunities are now popping up in the field of, in all domains of healthcare, not just computer aided drug design, but in all domains. And let me tell you that, I can understand that many of you over here do not have much knowledge of CAD, because recently, there were a lot of opportunities that were advertised related to computer aided drug design experts, basically scientists who can work in the field of CAD. And unfortunately, you can say, there was literally, um, a lot of dearth. Like, it was very difficult to find, you can say, students or professionals or individuals, who had some experience in CAD. So definitely, if you really get some good knowledge in CAD, there is a lot of scope in the industry for sure. I'm not saying it is not there in academia, but comparatively in the industry, the opportunities are much more. Okay. All right. So, let's talk about, when you enter the industry, how exactly do you kind of like contribute. So see, even medicinal chemists have knowledge of, drug design, of course. But then, what is the utility of a computer aided drug design, or what is the expectations? Okay, what are the expectations from a computer aided drug design expert, or what are the expectations from our artificial intelligence tools? So you might have heard a lot about, artificial intelligence, and how it is making breakthroughs in the field of healthcare. But from an industry point of view, what exactly are they looking for in someone who can contribute in computer aided drug design? Okay, they are basically looking for, like, for example, let's say we have to make 500 molecules. Okay, so if we talk about the hit generation state, okay, so from, to identify a hit molecule, let's say we have to synthesize 500 molecules. Okay, if we are not utilizing any tool, okay, let's say we are not using any artificial intelligence tool, nor are we using any computer aided drug design tool. Okay, so let's say we need to make 500 molecules. So as you can see, the time required is 1 to 2 years. Okay, so let's say we synthesize 500 molecules in this 1 year, and then we evaluate them, and we find some hit molecule. But if using the computer aided drug design, let's say from 500 molecules, because of computer aided drug design, we were able to identify the hit in 200 molecules only. So that is where the industry will find the relevance, that you have basically saved synthesis of 300 molecules, and most importantly, you have saved time. So instead of using 500 molecules, through the utility of computer aided drug design, you came to 200 molecules. Okay, so this is where you show your utility, or a computer aided drug design expert shows their utility in the field of drug discovery. Okay, so that is one aspect where you show your utility, right? So like I said, there are different domains, but majorly, you would be, most of the importance is given to molecular docking, and at the same time it is also given to molecular dynamics. Okay, so majority of the importance is given to molecular docking, because this is very, very fast. Okay, basically the advantage of molecular docking is that it is very, very fast. Okay, molecular dynamics is comparatively slow, and it requires a lot of resources. Basically, it requires high computational power. Okay, you need very sophisticated instruments, and also it will take a long time. Okay, so depending upon how long a simulation you are going to do, it might take a few days to sometimes months also. Okay, but molecular docking will happen in minutes or seconds. So it's basically a very, very rapid tool to give a preliminary idea of how the molecule is binding in the protein pocket. Okay, so like I said, this is like the small molecule and how it is going to bind in the pocket. Okay, there are different ways you can bind. So molecular docking gives us different basically ways in which this molecule can bind. Okay, and yeah, so that there are different domains like QSR is there. Okay, homology modeling is there. Of course, we will talk about these tools in the course. Since the time is a little short over here, so I'll not go into the details. Okay. So, like I talked about the 2021 breakthrough of the year, which is basically a tool which was developed by DeepMind, which is an offshoot of Google, and it's called AlphaFold. Okay, so you can see over here in the green over here is the 3D structure of the protein. So these green lines that you can see, the green ribbons, okay, the helix basically. These are basically the experimental results, and the blue ones over here are the computational prediction. So you can see it is almost perfect overlap. So, excuse me. So there's a perfect overlap of the experimental result and the computational prediction. All right. So this was a tool that was developed by by DeepMind. Okay, it's called AlphaFold. So this is the thing that I was talking about, which was called the biggest breakthrough in the field of science. Okay, now most importantly, see, we will look into CAD, and I'll talk about the different tools that we can utilize to to basically understand how the small molecule is binding. Okay, so basically in this course that we are going to start from next week, um, we would be basically going for a free tool which is called Autodoc. Okay, it is freely available, so all of you can practice on that. And then there's another tool which is called PyMol. Okay, so Autodoc is the software that is utilized for docking, and PyMol is called a visualizer, where you will try to see the structure of the protein and how the small molecule is binding in the pocket of the protein. Okay, so PyMol is basically a visualizer, and Autodoc is the tool through which we will do the docking. Okay, so see, docking and whatever dynamics, whatever tools I talked about over here, right? They all can be done by anyone. Okay, you can even tell a 5-year-old student or a 5-year-old kid also that you click this button, you click that button, you can give them a sheet, and they will also be able to do docking and dynamics. Okay, but in the course, we are not just going to do okay, click here, click there. That's not what. Of course, we'll be doing that how to operate the tool, but that you can learn from anywhere else also. The importance of this course is how to basically interpret. Okay, because honestly speaking, these are all done by tools over here. If you just talk about doing docking or doing dynamics or doing any of the other tools or any of the other domains, okay, whatever process you are doing, that does not require any brain. Okay, you can just randomly click and do the docking, okay, or dynamics or whatever it is. But to interpret the results and where to do docking, where to do dynamics, how to interpret the results, okay, how to know when the results are good, how to know when the results are bad. These are the things that you're going to learn. Okay, because even during the interviews also, let's say you do become an expert and you go for interviews in the industry, nobody is going to ask you how to do docking. They're going to ask you, okay, if this is missing, then what you will do? If that is missing, then what you will do? If you have 10 options, then which option will you choose? Okay, so these are the things that we are going to focus on during the course. That is the main aspect. Okay, anyway, so like I was saying, whom is it for? So of course, computer aided drug design, basically you should be fond of computers. Apart from that, you should like visualization. Okay, because visualizing the structure, okay, how it might be binding, looking at it, what kind of modifications you can do. Okay, so those are some of the things. Then of course you should be, I'm not saying you should know coding right now, I'm not saying that, but this is one very dynamic field. Okay, and let's say over the course of time, let's say in the next 3-4 years, you should, like I said, it's not just that you should have knowledge of just doing docking or doing dynamics. You should have a knowledge of medicinal chemistry also, and at the same time you should also have some knowledge of coding. Okay, that you will definitely inculcate when you will go forward, because coding is something, at least basic coding you should know. Okay, but even if you don't know right now, that is not a problem. Okay, you can start off with CAD, but I'm saying you will need to basically uh, um, improve yourself or improve your skill set or expand your skill set. Okay, going forward, you might need to know some medicinal chemistry principles, or some formulation principles, or you should have some idea of little bit idea of coding. So you need to, of course, expand your skill set. Okay, of course, it is for someone who is not a big fan of experimentation. Wet lab means basically being in the lab. Okay, so as a computer aided drug design expert, and especially in the industry, they require specialists. So basically, you should have a lot of knowledge, or you should be a specialist in the field of computer aided drug design. You will not be able to do some experimental studies. Okay, all of the things that you will be doing would be on the computer, right? So you should not be a big fan of wet lab, because after a couple of years it should not be like you are missing experimentation, right? In the industry, of course, in academia you need to do both also. Okay, so if you are a fan of wet lab also, and at the same time you need to learn computer aided drug design also, you can do that, but then you have to look for an opportunity in the academic field or in the universities. Okay, generally in industry, either you are doing experimentation or you are doing computer tools, you're utilizing computer tools, you're not doing both. All right. And of course, if you are going to full time be working in the field of computer aided drug design, you should not be someone who minds long screen times, of course, because you will have to work on a computer. So the amount of time you will be spending in front of a computer or in the front of a laptop would of course be very very high. So you also need to keep that in mind. All right. So like I said, what are we going to learn in this course? So it is basically how docking works. Of course, I'll be teaching you how to do docking, and everything of that. But apart from that, like I said, more importantly, what is molecular docking? What kind of things we need to keep in mind? How to interpret the results? How to take these results forward? How to make sense of these results? That is what we are basically going to learn. And then of course some very good visualization tools. So if you're someone who would want to even think about a publication also, or utilize molecular docking and then go forward with other tools also, so for them also it will be a great start to understand what exactly is computer aided drug design and how to interpret the results, most importantly. Okay, so that is something that we are going to learn in this particular course. All right. So thank you so much. Thank you so much for giving your valuable time. And I hope that you got an understanding of what exactly is computer aided drug design and what is its scope, and most importantly, how or what we are going to learn in this course that we have, which is starting next week, right? So over to you, Dharmesh. I think we can have a Q&A session, if there are any questions.
Surely. So, first of all, thanks a lot to you too for sparing time, and especially with regards to the kind of health situation we are all into. Special thanks for that. So, we have few queries in the chat box. First question from Abhimanyu Avasti says, with which application in CAD we can create DNA encoded libraries?
Um, okay, so Abhimanyu, if you can expand on this question, because the DNA encoded libraries that we have till they are more on the experimental side. So honestly speaking, I do not have an idea of how CAD would be useful in creating DNA encoded libraries. I'm not very sure. It's actually DNA encoded libraries. I'm good to like. It's great to know, or I will say I'm quite glad to know that you are aware of this field, because it's a comparatively new field, DNA encoded libraries. So I'm not sure, Abhimanyu, if actually CAD can be utilized in this particular, in the field of DNA encoded libraries. I'm not sure. So I might have to look into it, but I'm not really sure how we can utilize CAD in this field.
Thanks. The next question says, does learning CAD help only in the field of medicinal chemistry? And what are the different ways whereby research work in drug design of any medicine can be taken upon?
Okay, so yeah, I mean, CABs, CAD definitely helps in medicinal chemistry, and I think I expanded on this question, Sanika, that it can be used in different ways. So, for example, one is improving the potency of the small molecule. So, if you have a hit or a small molecule that is giving some activity against a protein, then how to improve the potency, then how to improve the solubility, okay, without altering the potency of the molecule. So when you go from, when we talk about lead
Sorry.
optimization step, right? So when you talk about the lead optimization step, you need to consider the solubility of the molecule, the toxicity of the molecule. If there is some toxic functional group, then you need to get rid of that. So that is where your computer drug design will come into the picture. Okay, like I had demonstrated earlier also, I'll just go to that slide to show you. So for example, let's say I want to improve the solubility. Now I know that this particular part of this molecule, it is going inside the binding site. So I cannot make any modifications over here to improve the solubility or to, let's say, improve the metabolic stability. But if I make some modifications over here, I know that this region is not very, very important. So if I make some modifications over here, I can improve the solubility. So that is just one of the parameters. Like that, toxicity is also there, metabolic stability is also there, like I mentioned earlier. Okay. Or let's say if you are making a molecule which is, let's say, something to do with the CNS system. Okay, basically it needs to penetrate the blood brain barrier. Then again you need to make a molecule which is able to penetrate the blood brain barrier. So again you need to optimize the properties. So basically CAD helps in first of all finding a hit molecule, and then secondly in what kind of modifications we can do to improve the different parameters. So that is where the CAD comes into the picture. And like you mentioned, what is the other question, what are the opportunities if we wish to do some research work in drug design? So like I said, most of the opportunities are right now in the field of in the industrial domain. Okay, in academics also there is, but comparatively the scope is less in academia right now. Okay, there's more scope in the industry. So basically industry hires, uh, you are basically equivalent to a medicinal chemist. So like each and every industry will have, or each and every organization in the industry, they will have different positions, right? Like scientist, senior scientist, leaders, group leaders, like that is there. So similarly like a medicinal chemist only, you will be hired as a scientist, but you will be working, on the, of course, on the which is called the dry lab. Basically you will not be working in the experimentation aspect, you will be working more on the like in silico aspect or the dry lab work. Yeah.
The next question goes this way. Are there any specific reference books for reading about CAD?
Uh, I have honestly not come across, you know, any great book as such for CAD. Most of my knowledge that has come from CAD is through research articles. Okay, and all of my colleagues or even my juniors and seniors, whomsoever I have interacted with, most of them have learned it through research papers and through practical work rather than through books. I don't think, this is my personal opinion, that more practical knowledge and more research knowledge would be advisable, because this field is changes. It's a very dynamic field. It changes very frequently. So books are really good for a fundamental understanding of the subject, right? But I think over here it is more to do with the applicability part. There are some very good book chapters. Okay, if you can just search, it's called, like, I'm not sure how we can, maybe we can write it in the chat section. It's called molecular docking in modern drug discovery. Maybe we can give the link also, Dharmesh, if that is possible. That is a good, you can say, starting point. It's a small chapter which basically introduces you to molecular docking and computer aided drug design. But as such, I'm not aware of any book.
The next question goes this way. Is redocking important?
I am not sure. I mean, if you can kindly expand on this question, Zahid, because I'm not. I don't know on from which aspect you are going. What you mean by redocking, actually. So, if you're talking about the reproducibility, so generally most of the tools that are used now, if you're talking about like docking them and again docking them to see if they're generating the same pose, then most of the tools right now are very, very accurate. So if you do the docking again onto the same protein, you will end up with the same, you will end up with the same pose. So redocking as such, I've not really heard of this concept, and we generally do not do that.
So re probably means kind of validation to check the robustness of the process or the software so that it's
Okay.
reproducible.
Right. Okay. Yeah. Of course. That of course comes when when you are if you want to do some validation, then molecular dynamics is the most important aspect over there if you want to do some validation of the docking. But what happens is, if the 3D structure is known, okay, if the 3D structure of the protein is known in complex with the small molecule. So let's say over here, as you can see in this slide, this is the 3D structure of the experimental structure of the small molecule bounded to the protein. So if you want to see the docking software whether it is generating this same pose or not, then you can do the docking, and if it is generating the same pose, then you can say, okay, the validation has been done, that the software is working fine, it has been able to generate the correct pose of the small molecule.
The next question goes this way: can we make triple helix plus a drug product by Abhimanyu Avasti?
Abhimanyu, like I mentioned, Dharmesh, as well, that DNA encoded library is something that I'm not very sure about how it can be utilized in the computer drug design expert. So as of now, I would not like to comment on that.
Next question from my end. When it comes to drug repurposing,
Yeah.
what is the first thing we usually target? Like, let's talk about COVID, where hydroxychloroquine was being set for prophylaxis, and other 2 3 molecules like doxycycline, ivermectin came into the picture. So, out
Correct.
of the entire drug library, what was it which first took them to that particular drug specifically?
So see, there are different ways by which even a CAD expert, like I said, should have some knowledge of medicinal chemistry, right? So if you see the few papers that did come up in for repurposing of of drugs against COVID, they were antivirals. Okay, so generally antivirals have broad applicability. Like if you, let's say, if you have made a drug which is used to treat some XYZ viral disease, then that can also be utilized to treat some other viral disease as well. So that was the first you can say that was the first you can say a process that was started, that take some FDA approved antivirals that are already present in the market, and let's do the docking against the proteins, the different proteins. So you might know that COVID has different proteins, like for example, the spike protein is there, which is on the surface, right? So initially that was the you can say the process that was started, that okay, let's take some antivirals and let's dock it to the spike protein or some other protein of the COVID, and see how the docking goes. If it is binding well, then maybe we can take them forward for the in vitro studies and in vivo studies, and then further to the clinic.
Nextly, when we talk about, sorry for being so much focused around COVID, but then I think there is a good chance over here. So just to understand, when we were saying that there were mutations in the virus, and we were evaluating different drugs, where initially we said one is working, second is working, then favipiravir came into the picture, and now the big word around molnupiravir. So is there any kind of dynamics possible whereby genetics, I'm again moving to DNA, but not with regards to our drugs being showing action, but if we can predict that these are the possible mutations, and if we have a drug which would be effective
Mhm.
against every mutation, then probably irrespective of the variant mutating, we can say that some same drug might still work out. Is that possible?
Yeah, your voice did skip in between, but anyway, I'll try to address what I understood is that, are you trying to ask that can we predict mutations beforehand?
Correct,
Is that
correct. Yeah, yeah,
yeah,
yeah.
that can definitely be done, right? But that would not exactly come in the field of computer-aided drug design. That will be more on the you can say predictive side, right? Okay, so if you have a general understanding of evolution and you know how the viruses mutate, right? So there's a lot of database that is already available. So this is, I think, some in this domain we can actually use artificial intelligence or machine learning, because if we have a lot of knowledge of how the mutations have evolved over time, then these kind of tools can also make predictions, okay, that what will be the future you can say mutations, and yes, based on that we can make some drugs, which we can also we can keep in mind that okay, even if these mutations happen, it still will work. So for example, like I was talking about, that when you make a small molecule, it shows certain interactions in the binding site of the protein. Okay, now let's say that binding site has 10 amino acids. All right. or let's say 20 amino acids. Now, based on some other tool, we have got to know that, let's say, arginine and aspartic acid, these two proteins are very, very important for binding of this small molecule. And based on some tool, some artificial intelligence tool or machine learning tool, we have been able to identify that this arginine is likely to mutate in the future variants. Okay, then we have to make a small molecule which is not dependent on this arginine. So that is one way that we can actually use computer-aided drug design to make molecules which are, you can say, resistant to mutations in the future.
Got it, sir. Before we move on to the last question, students have pinned a feedback form in the chat box. I request you to fill that form and we can proceed further. And yes, last question for the day. So, is there any book which provides knowledge of healthcare softwares like EHR and healthcare management softwares?
Again, Dharmesh, sorry to disappoint, but I am not really familiar of the books because, like I mentioned, I am more of a person who would prefer reading through research articles. So again, for even for any kind of software, not just for healthcare management or any other domain, for any kind of understanding or knowledge of software, I always prefer to go through research articles. If you want to know the utility or how to use these softwares, there are now journals, specific research journals, wherein they are titled methods. So in methods, what they do is they teach you all about how to utilize that software. Okay, so honestly speaking, I am a big fan of research articles. So again, I'm sorry, Neeraj. I think Neeraj has asked this question. I do not have knowledge of any book as such. But yes, if you are more than interested in reading through a book, maybe me and Dharmesh can find it out and get back to you regarding that.
Done, sir. And even I think we all would agree with the fact that our articles and our journals are super updated compared to the books which actually come up.
Of
Thanks a
course,
lot
of course,
for
of course.
entire discussion in, and yes, we will keep troubling you further. So students, we are going to start with the CAD course where we are going to discuss about PyMol Autodoc. It's going to be a course around 10 hours which would be spread over weekends. So if you at all you miss anything, recordings would also be available. And yes, this comes in with a hands-on training on the software which you have to practice at your end. The free versions, the demo versions of the softwares are already in place. So see you on the other side. The course commences from next weekend, and yes, we'll keep you posted. If you are interested for the course, just contact on the contact number being displayed on the ticker in the bottom or the one which is right being flagged over there. Thank you so much everyone for your time. Thanks sir for your time, and yes, please to take care. I hope we
Right.
could add value everyone, and yes, we'll keep you alert with further such sessions as well. Thank you so much.
Thanks, Dharmesh. Hope to see you all in the course, which will be starting off very soon. Thanks a lot.
Thank you everyone. Thanks.